So welcome everybody to the quarterly meeting of the California Cyanobacteria Harmful Algal Bloom Network. You can see the agenda on your screen. Thank you for joining. My name is Sarah Ryan. I'm the environmental director for the Big Valley Band of Pomo Indians and one of the co-chairs of the committee. Next year, there will be 2 of our seats that are open. So think whether you're interested in becoming a co-chair of this network. It's very meaningful and useful. There's a lot going on in the HABs world in California, as I'm sure you all know. I would like to have the other co-chairs introduce themselves. Go ahead, Jamie.
Yeah, hi, I'm Jamie Smith. I'm one of the co-chairs, part of CCHAB. And then I think, was Karen still on? Yes, awesome.
Oh, yeah, sorry, I'm kind of half on Teams. But hi, I'm Karen Odkins. I am at the California Department of Fish and Wildlife, and I am the newest CCHAB co-chair. So just learning.
Thanks, Karen and Jamie, for introducing yourselves. So you see on the agenda today that we are going to have a couple of presentations and also a work group report. I think we're going to be ending early. It's a lot of the items that we were potentially going to be putting on this October meeting have decided to wait until our January meeting. And the January meeting will be a 6-hour, or I'm sorry, it will go 9 to 2 or 9 to 3, depending on the agenda. And that was one of the announcements I wanted to make, is the dates for this year. Of course, I don't have those right in front of me, and I'm hoping one of the other co-chairs can provide the dates for the next CCHAB meetings. If one of you could go ahead and list that to the group. So our next meeting and then the other meetings of 2024.
Sarah, you got us on our toes. Give me 2 seconds here. I didn't have them in a formal list here, but I believe we had put the next meeting is January 31st. And then I can put, it'll take me a second, I can put the other dates in the chat.
Okay. Thank you for that. You can find on the CCHAB website, which hopefully one of the co-chairs can put that in the chat as well. But on the website, you know, as most of you know, there's been a transition to ADA compliant content on state sites. So there are, you know, the things are being transitioned over in a lot of different ways. Many things have been removed, but are still accessible. So, but on that website is our calendar. This information also goes out on the listserv that you can sign up for from the state to get announcements for all of the networks under the California Water Monitoring Council. And shout out to Nick Martano, who I see is on today. So thank you for joining us, Nick. All right, and are there any other announcements? I believe, Marissa, you had some announcements for that you wanted to go over.
Hey, Mommy, can I get some juice?
Oh, if you could
Good morning.
unmute themselves, that would be great. We have a couple people. I'm going to go ahead and mute folks just in case. Go ahead,
Sarah. Yeah, we were asked from some co-chairs to do a few announcements that's been going on. Some of the content was shared over the listserv. And so it's got some slides. Let me share them for you. All right. All right, so this is Marissa Van Dyke with the Water Boards, and I'm also here with Carly Nilsson. We co-lead the statewide California FHAB program across the state in close collaboration also with Department of Fish and Wildlife and Karen Odkins. And we just have a brief update for you. Let me go into PowerPoint mode. Okay. being a little slow. All right, so we just have update for the network. One of which is, again, with the statewide program, we implement it closely with all the regional water boards across the state. And one of the things that we want to note is that the Central Valley Regional Water Board now has a dedicated full-time coordinator again, Dana Schultz. We wanted to highlight that that position's been filled, and she's been with us over the summer. Thank you. And with the statewide program, we publish and host all the resources that are developed within our program and with other partners on the California HABs portal, again, the My Water Quality portal that Sarah was plugging. And that's where you can find our mapping tools, public outreach, the standardized voluntary guidance on how to respond to events of HABs and how to collect samples. And at any time you can ask us questions too. We have our general inbox heavily posted everywhere. So don't hesitate to reach out for any questions. So this summer, again, we hosted the pre-holiday assessment before the three major, summer holidays, Memorial Day, pre-4th of July, and Labor Day. We have a little summary here of the number of sites that were sampled along with our partners and along with the regional water board staff. And this year, because of the water levels, many sites were not sampled for around Memorial Day. There were still high water levels or cold water. So we are also hosting a post-holiday assessment to get some data in October. And so we'll be publishing all that data to our web map. We'll snapshot there is on the right. And we'll be presenting a summary of where, what the results were from the assessment at our January network meeting. So with the CCHAB guidance, the statewide guidance, that's posted heavily on the portal for both planktonic and benthic blooms and how to respond to those. The benthic guidance is up for review. It's been approximately 5 years. So the benthic HAB subcommittee that develops the guidance and then circulates it for peer review and proposes it for adoption by the network has been reconvened. We started just meeting in May of this year. We paused over the summer, and we'll be meeting again in a couple weeks in October. We'll be meeting monthly, doing quite a bit of homework reading and having quite a bit of presentations to review the most recent science and consider revising the benthic guidance for the following year. We don't have quite the timeline yet. We'll be developing that shortly and letting the CCHAB network know if we'll be proposing revised guidance for the following year. If you're interested in participating, even as a reviewer, not necessarily have time to meet with us on our monthly meetings, feel free to reach out to us. Also, pretty cool event that's happening starting this month is the USCPA Cyano Symposium. They're hosting 4 individual days across October, and there's going to be quite a bit of updates on what EPA has been working on with their partners. And also each day there will be a different state highlighting different elements of their HAB programs. So it'll be really beneficial to see what other states are doing in different aspects. On the last event, California will be highlighting part of our program, particularly our illness work groups, since we get quite a bit of questions from other states on how we're doing this and how we're reporting to the CDC. And so we got some more information here for you if you want to check it out. And lastly, we wanted to give you an update, just in case you didn't see the listserv announcement, that our satellite HAB map has been gotten an additional functionality added. The satellite tool also presents now the chlorophyll A data that's remotely sensed, in addition to the cyanobacteria abundance that was already there. It's a separate kind of toggle view of the largest lakes in California. So we just want to kind of give you a quick demo of that if you're not too familiar with it. Let me just leave this for you so you can go to it. Great. All right, so from the California HABs portal, you can get to our satellite tool, and this is the opening screen. And the disclaimer is quite useful right now for everyone new to seeing the chlorophyll A data. It has a plug for that, how it's in beta version now. And it's presenting the remotely sensed data from the Sentinel-3 satellites, the same satellites that are presenting the cyanobacteria data on this tool. And we updated that in July of 2023. Along with the disclaimer, you can go to the instructions, and the help instructions toggle has a little bit more information about what algorithms being used for the chlorophyll A data, and how to view it a little bit more with some more screenshots on how to view it with our statistics. All right, we're going to exit out of that. So when you first go to the tool, it's going to default on what we are familiar with seeing, the cyanobacteria index, which is shown here on the legend. You can change what you're looking at by toggling here from the cyanobacteria index to the chlorophyll data, and it'll show it'll change the statewide view when you do that. In addition, we are also presenting 1-day data. So the individual days when the satellite goes over California, you can see that individual day, in addition to what we've always been presenting, that 10-day composite, or kind of somewhat an average over the 10 days. Because satellite doesn't necessarily go over every day. There's gaps in data, and it makes the data quite choppy. So it's a little easier to view the data, and particularly use it for incident response and field assessments, that kind of thing. All right, so you can switch to the chlorophyll A data. And so the statewide view changes a little bit because we're also showing the near shore ocean data. And you can view it here in this statewide view. You can go straight to a lake, just like you do with the cyanobacteria. And it's going to default the graphs here in this first tab on chlorophyll A data, and you can see the trends over time. I'm right now on the 10-day data. And you can toggle here while you're in the statistics to the 1-day data. And so you can see here on the timeline, you can see the choppiness, and also in the most recent data. So you can look at it in different ways. We recommend starting with the 10-day data. And then many of our partners would like to see a comparison of the chlorophyll A and the cyanobacteria data. And we just caution that, the units are very different of remotely sensing these parameters in the surface waters. And so, have that kind of consideration when you're looking at your data. The units are very different, and they're going to scale different. So, you can look at some general trends. So we did make comparison graphs where you can compare the cyanobacteria index, which we're very used to looking at, with the chlorophyll A data on different axes. And again, very different units, different scale. Cyanobacteria index in the log scale, and is measuring, if they're not measuring equivalent parameters again, but you can, it is useful to see some trends, and you can toggle between seeing for that statistic, either the 10-day composite or the 1-day. All right. So just wanted to give you guys a demo. Again, you can zoom out back to your statewide view. You can toggle here again to what you're looking at, and also here you can toggle which data set. And we're considering this chlorophyll A data as a rollout, just kind of like a beta rollout. Or welcoming feedback from partners that use this and how useful it is or not. Any questions that you have, and we'll work to refine how this data is presented over time. And thank you for letting me do some announcements. Do we have any questions? I don't have the comments open.
Thank you,
Okay.
Marissa.
I'll take it as none. Okay, I'm going to stop sharing. Thank you.
Thank you for that. Very interesting. It's so exciting to see the chlorophyll A data statewide. All right, I think are there any other announcements for the group? Otherwise, let's go ahead and move into our next agenda topic, which is regional updates. And let's go ahead and start with Region 1. I see both Michael and Rich on the line.
Yep, can you hear me okay?
Hi, Mike. Yes.
Great. I'm going to share my screen. Okay, can you see that? Awesome.
Yes.
Okay, good morning, everybody. My name is Mike Thomas. I am the FHAB program lead for the North Coast region. I work alongside Rich Fadness, who is our swamp lead. Rich and I were pretty busy this summer, so I'm going to try to go through this pretty quickly, though. So in the North Coast region, we had our typical routine monitoring that occurs on Big Lagoon. This is done by Big Lagoon Rancheria and US EPA. They are doing biweekly microsystems monitoring. Similar in the Klamath Basin, we have the Hoopa, Karuk, and Yurok tribes coordinating with Pacificor and US EPA to monitor for microsystems. Also in the Russian River, Sonoma Water and Sonoma County are doing visual assessments in cyanobacteria ID. And to the right there is just a picture of the stations throughout the Klamath Basin. For pre-holiday assessments, we did 10 water bodies. You can see when these were sampled and the resulting postings listed in the table here. We will be participating in the post-holiday assessment as well, like Marissa described. We also did a partner monitoring project with Big Lagoon. This was in addition to their biweekly microsystems monitoring. We were having them collect a sample for the cyanotoxin suite and cyanobacteria identification. On that, in the graph at the bottom there, we have microcystin or all cyanotoxin concentrations over time. You can see that throughout the season, a lot of the microcystins concentrations were leading to both caution and warning postings. In addition to that, we documented 6 cyanobacteria genera throughout the season. Region 1 also did a special study, benthic monitoring in rivers. In this study, we were evaluating and implementing the tiered approach that we recommended in 2 reports that came out last year. This includes deploying SPATs as sentinel samplers, then conducting visual assessments to see how much benthic cyanobacteria is present, and then collecting mat samples to confirm toxicity. We were doing this weekly and 4 rivers from mid-May through October, and next week is our last sampling event. These results will be reviewed by the CCHAB benthic subcommittee when we're revisiting the guidelines this winter. We also participated in some US EPA studies, the ROAR project, which is developing standardized benthic sampling protocols. Basically, what we did this summer was a lot of transect and disturbance sampling in the South Fork Eel River. We are also doing some RNA and DNA research. We are using paraphytometers to determine the upregulation of genes at different growth stages. So essentially removing a slide every couple of weeks or so to see what's present. We're also doing some metagenomic sequencing for the ANA-F genes to characterize various anatoxin producers. Some other research, we were collaborating with the University of Nevada at Reno and CSU San Marcos, looking at spatial heterogeneity of benthic cyanobacteria and cyanotoxins. This was occurring both in the Salmon and South Fork Eel rivers. We also had an opportunity to jump on a helicopter flight and check out the South Fork Eel River. And what we were trying to do was see how readily observable microcoleus mats were in the river. You can kind of see in that picture to the bottom right there that they were easily observed from the helicopter. So this is something very interesting. We also conducted a lot of trainings in 2023. We hosted the US EPA ROAR training here in Santa Rosa on the Russian River. We also did some tribal trainings with Blue Lake Rancheria and Wiyot tribe. And then we held our partner trainings, which had a virtual and 2 field components in late August. And just to sum up the postings that we had in the North Coast region, you can see the various benthic and planktonic and the number of postings there. We also had a marine bloom this summer in Humboldt Bay, which was interesting. As always, we'd like to thank our partners. They are very much important in the work we do. So with that, I think I will stop sharing. Thank you.
Thank you, Mike. Appreciate that. All right. Any questions for Mike or Region 1?
Yeah, I was curious, your marine bloom, Mike, was that a marine species blooming, or was it transfer of like a cyanobacteria into the bay?
It was marine species, and I'm blanking on the name right now, but it was non-toxic. So basically
Okay.
the caution was listed when it was present, and then Humboldt County took it down after that.
Interesting. Thank you.
Mm-hmm.
All right. So no other reports for Region 1. Let's go ahead and move into Region 2. And I think I see Rebecca.
I think actually Shannon had a question perhaps.
Oh, I'm sorry. Shannon, go ahead.
Hi, good morning, team. Just wanted to, give kudos to Region 1. They've done a lot of yeoman's work this summer. So thank you so much, especially on the benthic front. That remains one of our big frontiers in the HAB world. The event at Humboldt Bay was due to Noctiluca. It is not known to produce a toxin. It is a marine-based organism, and it is known to be tomato soup in color. It is a high ammonia producing bloom, so it can be quite irritating at its peak because it can aerosolize ammonia-based compounds that are very irritating to the respiratory tract. If you were to put any of the algal material, dried algal mat type stuff into your mouth, you can have, or a dog was to ingest it, you could have secondary irritation of the GI symptoms. And in fact, we did have a dog case that we determined was HAB related. So again, one of those kind of different scenarios. This is really a new one for us to see this, but it is known to occur in marine environments. They tend to be fairly transient. They tend not to have a big impact on wildlife because wildlife in general can leave the area. The blooms tend to fade very quickly. But again, because they can produce that ammonia, if there's dried material on the beach that then children or dogs, for example, get into because it's fun and stinky and different, then you can have some GI symptoms or kind of dermal or mucous membrane irritation symptoms. So anyway, thanks so much.
Thanks, Shannon, for that additional information. And Marissa, I noticed that you put the species in the chat.
Just in case folks weren't familiar with the spelling, it's a little bit more of a rare marine bloom species.
Great.
And there's also a link to an article that we referenced. Washington State seems to experience these more orange blooms, and they have some targeted outreach for the public about that to stay away from them as well because of the ammonia.
Thanks, Marissa. All right, Region 2, Rebecca, you're up.
Hi, good morning, everyone. I'm going to keep my update really brief since I saw most of the agenda items are about things that were happening in my region. So this what we've been up to this year. We did the holiday sampling. We've had a few incidents to respond to, majority of which were red tide related. And then of course, East Bay Regional Park District always does their weekly monitoring, which we're really thankful to have them as a partner in that. And I guess one highlight was we did have a dog illness that happened at Crissy Field Marsh. So it's the marsh that drains into the beach in Crissy Field in San Francisco. We sampled there and there were cyanobacteria present in the sample, but no toxins detected. And the dog recovered, so that's great. And we're currently investigating a report of dead fish at a lake in Sonoma County. That just came in last week. And we also had a few reports come in this year that ended up not being sound related. So definitely the most activity has been marine related, mostly in the San Francisco Bay. But we did also have a marine game in Drake's Bay, which is Point Reyes National Seashore. And then we also had a report of sort of a transient red tide situation that happened kind of off the coast of San Mateo in the Pacific Ocean. Our holiday assessment, we sampled 5 different areas across the bay, and 2 of them, a marina lagoon and another in San Mateo, and then a Berkeley Aquatic Park, which is also a kind of weird lagoon system in Berkeley, both warranted caution signs. And then I think one of the most important things that happened this year was some expansion of our partner monitoring. So we expanded on a partnership we developed with the Rotary Nature Center Friends at Lake Merritt last year, because the red tide that happened in the bay last year moved its way into Lake Merritt. Lake Merritt is in Oakland, it's connected to the bay. So we developed a partnership with them. And so beginning this year, in May through October, they're collecting 2 samples monthly. And then state board and region 2 are splitting the cost of paying for those lab analyses. And thankfully, so far, we haven't seen any cyanotoxin above caution advisory. So there's been no advisories for Lake Merritt this year. And the red tide also did not make it into Lake Merritt this year, so that's great. And we also did 2 workshops with environmental organizations in the North Bay. So Napa iCare, which is the Napa Institute for Conservation Advocacy, Research, and Education, and the Sonoma Ecology Center in Sonoma County. So we did trainings with them. They, the community comes to them asking about HABs. They're boots on the ground. They see what's going on a lot more than we do. So we provided them some training and equipment. So if there is an incident, they can help us respond to it a lot more quickly. And that is super important because Christina and I only have about 5% of our workload is responding to HABs. We don't have dedicated HAB resources in our region. So we're super thankful to have partners to help us respond to things when needed. And that's all I have.
Thank you, Rebecca. Are there any questions for her? For Region 2? All right. Thank you very much for that. All right, we'll move into Region 3, and I don't see Melissa. Marissa, are you going to go ahead and report for Region 3?
Hey, yeah, I can try. Also, Curly, if you want to weigh in Region 3. Did we get a write-up from Melissa Daughtry for the Central Coast area?
No, but
so. Okay.
for their highlights, they did sample 10 different water bodies for the holiday assessment. It's part of their regular monitoring program. So once again, they're one of those regions that don't have dedicated funding. So they really try to leverage some of their existing monitoring efforts. And they did have some, and I'm not going to recall the water bodies, I didn't pull it up. They did have some danger advisories, which has been pretty consistent in San Antonio. And then the river and the lake. I'll put it in the chat.
Yeah, Oso Blanco had advisories as well. Right.
Okay.
And with their pre-holiday assessment, even before that in May, there was concerns about drinking water contamination in Lake San Antonio. So we were doing some ambient monitoring to set the surface to try to do some surveillance and support the water managers. And then the response that was handled by the Division of Drinking Water and the local health agency. We normally don't post if there's a drinking water advisory on these maps, but we can share that information separately. Are there any other updates for the Central Coast areas? That's San Luis Obispo, Monterey Bay, down to just about Santa Barbara.
Marissa, I do have a question for you if there's no other updates for Region 3. So the drinking water advisory for, you said it was Lake San Antonio?
Mhm.
So is there tap water, cyanotoxin testing happening there, or was it just that there were high levels? Are these private water systems, self-supplied water systems on the lake? How did that happen?
Right. So there's a public water system that draws from Lake San Antonio, and they noticed something going on earlier in the year, normally before we started with our holiday assessments, and that concluded after about 3 months. They were monitoring for cyanotoxins and putting out "do not drink" when appropriate. The toxin levels just seem to get treated and with removed from the water systems. We're a little bit disconnected on how on the notification process because the drinking water advisories go through another department, another agency. But we were monitoring just at the surface, and so there were recreational advisories going on at some of the beaches for Lake San Antonio as well, because there's some accumulation along the beaches. I can follow up with you separately on some more of the details about how the drinking water monitoring went.
Thank you. That'd be great because that's definitely one of our programs here on Clear Lake.
Right.
Valley.
Yeah, sorry. Yeah, I don't have those details too handy because it was much earlier in the year. Apologies.
Great. All right. Thank you. Any other questions for Marissa or Central Coast region? All right. Moving to Region 4, Los Angeles. I didn't see Emily, or is there anyone else from Region 4 who can report, or Marissa or Carly?
The map does not want to move anymore. I don't believe Emily provided us a summary for this year, and they did not participate in the holiday assessment. Sorry, there's a little issue happening with the map. I need to refresh it. All right, let's go back to the LA area. For the LA area, there are some advisories currently, and I think we have Brianna from the Department of Water Resources, so I don't want to supersede her summaries, but there's some advisories for Castaic Lake, and there was one for Pyramid Lake. I'll defer to the Department of Water Resources to go over that material.
Brianna, you want to jump in for that? DWR report? Or we can come back to that too.
Okay.
Anything else for Region 4 at this point for water boards? All right, let's move to Region 5 with Matthew on.
Hi, this is Dana, and I can do
Dana,
Region
I'm
5
sorry,
today.
Dana. Yes.
I have a couple slides that I was going to share. Works. You might be seeing the wrong screen.
Yeah, so just go
We're
up
seeing
to
your
the
speaker slides too.
you can, if you go back on that, if you present, you can just switch displays. So see that middle toggle that says display setting on the speaker side, just say swap. Yeah, there you go.
Okay,
Perfect.
how's that?
Perfect.
I don't know if I started, might have lost my access to turn on my camera. I can just do it like this. Okay, Good morning. I'm Dana Schultz. I started this summer with the Central Valley Water Board. And I was going to give an update today about our main partner monitoring program, holiday assessment, and the advisories in our region. So this summer we worked with Restore the Delta in the Stockton area, and they went out and sampled 7 sites along the Stockton waterfront and San Joaquin River. They were going out monthly to collect samples to send to Ben Genetics for microcystin analysis and a suite of toxin genes. And they're also going out weekly to test with the microcystin strip tests. The most recent Ben sampling was the week of 9/18, and results came back with no microcystin nodularin detection, which has been the case for almost every event this summer. And there's one additional Ben sampling event planned for October. And then for holiday assessment, the portion of the events that Marissa mentioned that happened in Region 5, there were 2 sites sampled at Hensley Lake that ended up with warranting lake-wide caution advisory. There were 2 sites sampled at Eastman Lake and no advisory was recommended. And then Sarah Ryan and Big Valley Band of Pomo Indians sampled 6 sites, and that I believe is with their data set. And then we do have plans for some post-Labor Day sampling, as was mentioned, especially at Kuya River. The water levels and conditions are such that they were not, sites were not accessible until recently. We just got results in from those samples and no toxins were detected. And then we also have plans, Akira with the Fresno office is going to sample Hensley Lake in the next few weeks. And then advisories currently in Region 5. So I have it broken down by advisory level and lakes where those advisories were present. So we have danger advisories at Clear Lake and Contra Loma Reservoir, a warning advisory at some sites in Clear Lake, caution at San Luis Reservoir, Lake Isabella down near Bakersfield, and Clear Lake. We have a few visual bloom observations that do not have accompanied toxin testing right now at Old River, Middle River, Turner Cut, and Holland Cut. They're all kind of in the Delta region. And then a couple algal mat alerts, one at Clifton Court Forebay, and one from state park sampling at the North Fork American River. And I believe all of these at the moment are mostly from the routine monitoring programs happening in our region. And that's it.
Thanks, Dana. Any questions for Dana? All right, I'll go ahead and give a Clear Lake update real quick. Let me share my screen. So we continue to over at Clear Lake with Big Valley Band of Pomo Indians. We have about 20 sites, including some sites that we're getting in the interior of the lake from the county with their interior of the lake monitoring that they do, water chemistry and other things, and also UC Davis, who is out there doing various studies. So those, along with the shoreline sites that we have, we've been doing monitoring twice a month throughout the summer. And so you can see the a spreadsheet that is basically identifying the caution warning danger levels. Most of this is all of this is microcystin. And we make recommendations to the county public health officer in terms of signage. We are continuing to see microcystis and other microcystin producing cyanobacteria, because we do microscopy for dominant and secondary genera on at each of our sites. We've got a really great documentation of of this data. which will be helpful. The toxin levels this year were a lot less than a couple of years ago, which is great. I think the highest toxin level we got was for microcystin 3,059 micrograms per liter at a site in the lower arm of the lake, which is usually where we see some of our highest. In general, we were mainly seeing throughout the season, switch over from nitrogen fixing cyanobacteria. We were starting to see that. We were seeing that in the early part of the season, but it switched over to non nitrogen fixing cyanobacteria, mostly microcystis through, as we got further in summer. We have been conducting some toxin analysis in the creeks as well, and putting that on our website, and doing benthic cyanobacteria analysis, as well as some spat bags. Here's our website, bvrancheria.com/Clear Lake cyanotoxins, with the most recent data that we have analysis for. Nice thing about our site is you can see some of the microscopy photos and site ID photos. So in general, we've had, it's been a relatively good year in terms of caution warning danger levels compared to previous 2 years, but still a number of, I believe there were some dog illnesses and a confirmed dog death, and some human illnesses on the lake that were confirmed from the illness work group, and we continue to do outreach with the public on these issues. Any questions? Any other Region 5 report out? Yeah, go
I had
ahead.
a question for you. This is kind of like, it's okay. Like, I'm curious with your experience on the lake now over several years. Do you feel like with the big rain season that we had, are you starting to see that maybe like if you see more rain, you see less high intense toxin events, or don't know yet?
Well, the toxin levels this year were higher than last year.
Okay.
So yeah, we were kind of wondering about that, but last year when we were still in the drought, we were expecting, in the summer, we were expecting to see, I guess, the last 3 years, the highest toxin levels, because we got up to 160 micrograms per liter for microcystin in 2021. And then, 2022, the drought was even worse. We did not see toxin levels, I think, breaking even 10,000 micrograms per liter for microcystin. Heavy rains came. We thought, oh, well, the lake level is high. Maybe we'll see, we won't see things, but as high, but we actually saw them, toxin levels were higher this year compared to last year.
Interesting. Okay, thank you. I was just curious.
Yeah. Yeah, you're welcome. All right, thank you. And let's move to Region 6. I believe Sabrina is on the call.
Hi, good morning, everyone.
Morning.
I'm Sabrina Rice. I'm the harmful algal bloom coordinator for our office. And I have a slide here I'll share. Uh-oh. Oh man, I've been having can you guys still hear me?
Yes.
I've been having so many computer issues since my computer was updated. So everything is frozen. But I'll just wing it.
If you want to email something over, we can share it real quick.
Yeah, I can't even click on my screen.
Okay.
So yeah, I'll just make it, I guess, a little brief. So we participated in the pre-holiday assessments this year. We also did the post-holiday for most of our water bodies, just because we had such a high winter or heavy winter. And then a lot of the past data that we had, we weren't seeing a lot of hits during the Memorial Day assessment. And we sampled roughly 20 water bodies for each of the assessments. And then we also tried to start a new routine monitoring program this year with Inyo County. They have been sampling Diaz Lake, Mill Pond in Bridgeport, and when they could get to it, they sampled Pleasant Valley Reservoir. And then we had like 3 danger advisories this year. One was at Indian Creek Reservoir in Alpine County, and we had Crowley Lake, which we actually issued a lake-wide advisory for. We used some of the satellite data to kind of help us make that decision. And then at Silverwood Lake, we had 2 danger advisories. That one has gone up and down, and currently it's back at a warning. And then we had a few illness reports this year, and off the top of my head, the only one I'm remembering that was actually confirmed to be HAB related was at Reagan Beach in Lake Tahoe. And yeah, that is it.
Thanks, Sabrina. Do you, are there any questions for Sabrina or Region 6? And, in the January meeting, all of the regional HAB coordinators will get a chance to, fully go into their year and have slideshows and things and more time to do it. So thank you for that. All right, so we'll go ahead if there are no questions. Oh wait, looks like Christine has a question here for Jamie's question. Is this related to Region 6, Christine? This is for Jamie's question. I wonder if the runoff from the watershed is still high in nutrients. Oh, are you probably talking about Clear Lake?
Yeah, I was talking about Clear Lake.
Oh, sorry. Still high in nutrients.
Yeah, with all the forest fires that have been going on over the past couple of years during the drought, I was just kind of curious if whether or not, the runoff coming off during high rain events and wet years, I'm wondering if that's still having an impact in the downstream water bodies. I just didn't know if there was any studies sort of looking at that.
I think the county has some information about phosphorus loading after the fires, because we had the 2018 Mendocino Complex fire being one of the big ones. And they were not, I believe they were not noticing that phosphorus was increasing. So, but I can also have ask them to do a presentation at one of the next meetings on that issue, because they have been doing some work on that.
Awesome, thank you.
Yeah, thanks for the question. There was also a question I noticed in the chat about where can we get, so this is Jenna Lasco. We encountered folks swimming in also Flaco Lagoon, sorry if I'm mispronouncing, when we were conducting Tidewater Goby surveys in September. Is there a place where we can get signs for the public? And I believe, Marissa, they just need to, should they go to the state or to the region for that?
Yeah, they can email our CCHAB reports email, and we'll route them to the right person who has some signs for that area.
You, Marissa, are there also benthic signs?
Yeah, benthic signs are available as well.
Okay. So thanks.
Just a little less often. You
Yes.
can order as many, but yeah.
Okay, thank you for that. Okay, if no other questions for Region 6, we'll move to Region 7. I didn't see Jeff or someone from Region 7, or is there somebody on the line who can, or Marissa, who can go over that report?
I can take this one on. So this would
Thanks,
be
Carla.
for Jeff Geraci, and they did participate in the holiday assessment. He went out to the Salton Sea. They did have a caution advisory in the heat in the middle of the summer around the 4th of July. And then they also did perform some holiday assessment, like incident response monitoring for San Bernardino Parks at Weist Lake, which also did have a caution advisory. And then as a side note, he's actually working with a group with the US EPA, and he's going to be traveling up to Oregon, I believe, to work with the tribe and the state of Arizona on some like Havasu kind of starting some discussions on how they can start addressing HABs there. So that's pretty cool and exciting stuff.
That's great. Thank you. Are there any questions for Carly or on Region 7 or any other reports for Region 7? All right, moving right along, Region 8, Riverside. I didn't think I saw anybody for Region 8.
I can take that one too. They also participated in the holiday assessment this summer, and they sampled a couple sites at Big Bear Lake and Mill Creek, and then Lake Hemet, sorry, and Lake Elsinore, and they had caution advisories for all of them. So the good news is for Lake Elsinore is it stayed pretty steady at the caution advisory. And they have done some quite a bit of work setting up collaborations with the city. So now we actually receive their data. They collect samples generally every 2 weeks. So we've been doing a lot of data sharing to make sure that our HAB map is reflective of the current conditions.
Any questions for Carly on Region 8 or other monitoring? All right, moving to Region 9, San Diego. I think I saw Carrie on the call.
Yeah, hi. And I do have just some quick slides. Let me see if I can share this properly. Let's see. Where is... All right, let's see. Is that working better?
Yeah, looks good.
Okay, great. Yeah, so this one's really brief, and I didn't want to repeat anything from my August update, but I didn't have a slide, so I'll just talk about the holiday assessments. We did 6 high recreational used streams this year, and we didn't have any detects until the very last one in Labor Day, and pretty low level toxins, but we did have caution and then just alerts. And one of them was pretty neat. We were able to give thank you the durable signs to it was an HOA and developer near the lake where we had that one. It was a ponded area of a stream. So it was 3 out of 6 that ended up having any kind of warning this year. And then just currently the blooms that we have right now are mostly at lakes. So Diamond Valley Lake, that is Metropolitan Water District managed. And that's been at a caution ever since July 5th for microcystins. The warning's still on their website as it's shown there. Lake Morena, that one has a bloom that started at the end of August, and that also is high for microcystins. And then Lake Henshaw, it's just posted for caution year round. And the last, they do weekly monitoring, that's Vista Irrigation District. And right now they actually aren't detecting toxins, but they just have the precautionary sign and it's posted on their website too. And then finally, a smaller county park lake that has had a bloom since July 7th, and that one's also microcystins. And then also this year we did 2 special studies, and I'll just focus on the second one. We had finished the first one that I talked about in August. This one was a continuation of a study in a high agricultural use watershed. It's the Santa Margarita River watershed. And so it was a follow-up to a past study where we deployed spat, and we got pretty decent concentrations in the spat, but we didn't have mat or water samples collected during that study. So this year we wanted to try to pair them so that we knew what those spat results actually meant. And good for the stream, bad for our study. We did the spat deployments in late July through August, and we got mostly non-detects or very, very low, nothing that compared to our study before. So we paused for a little bit and we did one last deployment, and that was in late September. And we're still just waiting the results from Ben, so we'll see what happens with that one. And that's really all I wanted to say for this meeting, so thank you.
Thanks, Carrie. Any questions for her or for Region 9?
Let's see.
All right. How about other entities, State Department of Water Resources, or anyone else, any other entities that are doing HAB monitoring on the lake or in the state that they'd like to report out? All right. All right, well, let's move to our next item on the agenda. It looks like we're pretty much on time, which is great. And this is a presentation with Keith Bomer-Gregson from USGS and Dave Sen from San Francisco Estuary Institute on the 2022 Heterosigma bloom in San Francisco Bay. So if you guys would like to pull up your presentations, and you have until about 10:25 with questions. And Keith, would you like questions throughout, or would you like us to hold questions till the end?
Holding questions to the end would probably be helpful.
Okay, thank you. Will
Great.
do.
Thanks. Yeah. Okay, is the screen sharing working?
Looks good.
Okay, great. Also, in the course of this meeting, I've gotten dropped off the call. I think this might be our office network. So I apologize if that happens, but I've usually bounced back. So but hopefully I'll make it through. Yeah, so hi everybody. My name is Keith Bomer-Gregson. I'm at the U.S. Geological Survey and the California Water Science Center based out of Sacramento, California. We do a lot of our work in the Sacramento, San Joaquin Delta and San Francisco Bay. And today I'll be talking about the Heterosigma bloom that occurred in San Francisco Bay last year in 2022. And this has been a close collaboration with the San Francisco Estuary Institute and Dave Sen and the water quality program that he leads there. And so we put together these slides collaboratively. Actually, a lot of the slide content is from Dave, but I'll be doing the presentation today. But Dave is here and will be helping answer questions at the end. And also want to talk about some new funding we just got from NOAA through their MIRHAB funding program. And so I'll, after we talk about the bloom, we'll talk about the MIRHAB project and share that information with you all, and then can take questions collectively about everything at the end. All right, so first off, the bloom response and kind of this work on harmful algal blooms in San Francisco Bay has been a very collaborative effort. San Francisco Estuary Institute, Dave Sen and the Nutrient Management Strategy. They've been one of the chief funders and folks focused on water quality monitoring the Bay for almost 10 years. The USGS Biogeochemistry group that I'm a part of, we do a lot of the field work and data collection for Dave Sen's group. And then there's a lot of other people involved as well. Rafe Cadella at UC Santa Cruz, Tim Otten at Ben Genetics, San Francisco Baykeeper, San Francisco State University. So really just it's a lot of this work, and especially the bloom response last year wouldn't have been possible without a lot of people chipping in their time and their efforts. And particularly the funding too, a lot of this comes from the Nutrient Management Strategy. We've received some internal USGS funding, and we're also able to receive NOAA HAB event response funding for the event last year too. And so San Francisco Bay is the terminus of the San Francisco Estuary, which begins where the San Joaquin River and the Sacramento River meet up in the Delta, and then they flow out the Golden Gate. And the bay itself is highly enriched in nitrogen and phosphorus, which the majority of those, the source of those nutrients coming from wastewater treatment plants, which you can see here as green circles on the map. It's within the 90th percentile of estuaries in terms of nutrient loading globally. And about 10 years ago, the wastewater treatment plants and the regional water board came together to create the San Francisco Bay Nutrient Management Strategy, which was tasked with investigating the question about the impacts of nutrient loads to San Francisco Bay, and if those loads are having an adverse impact, and what management actions would be needed to prevent any current or future impairment. And so SFEI has been the manager of that nutrient management strategy program since then. And the key stakeholders in this effort are the regional and state regulators, the US EPA, the wastewater treatment plant dischargers, the fisheries agencies, NOAA, NIMS, California Department of Fish and Wildlife, the Ocean Protection Council. There's numerous NGOs involved, as well as the water resource management agencies. And so to boil down, the kind of key question is, does San Francisco estuary have a nutrient problem now or in the future? And then how could these impacts be mitigated or prevented? And it's quite there's a big price tag for this. If large-scale wastewater treatment plant upgrades across the bay to reduce loads are on the order of multiple billions of dollars required. So everyone is wanting a firm science foundation and understanding about the rationale for why management actions would be required, and also the levels that would need to be targeted. And historically, the San Francisco Bay has been resistant to eutrophication and high algal or phytoplankton biomass. The reason for that is that the bay has been considered light limited, and so nutrients have often been in excess. So it's not so much that the nitrogen or phosphorus are limiting growth, but more there's not enough light to provide the energy for more growth. And so these three mechanisms have been the main limitations. So one, that high turbidity, which reduces the light. There's also strong tidal mixing, which mixes the phytoplankton into deeper water. And then there's also a large amount and high biomass of filter feeding clams. So they are so grazing and top-down controls are also high in the estuary. But over the last 10 years, there's been thoughts that dynamic might be changing and the bay might be becoming more sensitive to nutrients and maybe respond differently than it had in the past. So as a result of these programs, there's now in place quite a number of HAB event observational resources. The USGS has been monitoring in the bay since the late 60s, early 70s, and much of the funding now is provided by the Nutrient Management Strategy and SFEI. So there's monthly cruises that go up the spine of the bay. There's also SFEI has installed numerous in situ water quality moorings, providing data every 15 minutes for chlorophyll, pH, dissolved oxygen, and other sensors. We also, the biogeochemistry group that I'm a part of, we have on our boats, we have a mapping boat that has a suite of sensors and a flow-through system, so we can drive around at 25 miles an hour, constantly pumping water up and through our sensors, and everything is stamped with a timestamp and then gets GPS coordinates. So we're able to generate these maps showing where we've been and what the water quality looked like in those areas. And so we're able to provide this high spatial coverage maps through that. And then we also have remote sensing, citizen scientists, and numerical modeling also being done in the region. So that's kind of the background for San Francisco Bay. And so prior to last summer, there really had not been any widespread harmful algal bloom event. So in spite of a lot of effort, been working on the bay and monitoring the bay since the late 60s, no large bloom had occurred with major harmful impacts. But that all changed last year. So in late July 2022, the first observations about discolored water started to come in the Alameda, Oakland area here. And it was determined that organism was called Heterosigma akashiwo, which is an ichthyotoxic or a fish toxin organism. So it doesn't really have, there's not human health impacts, but it is toxic to fish. And so then we started tracking this with remote sensing imagery. And as you can see here, kind of in these early days in early August, high chlorophyll, which is represented by these brighter colors, was initially constrained to this Alameda area. But then around August 10th to the 20th, it began to expand into the more open waters of the South Bay. And then in particular, it really took off from the 20th to the 25th. That's when biomass got its highest, and it pretty much encompassed the entire South Bay. And then it very rapidly crashed and decreased around August 30th to 31st. Very large impacts. Chlorophyll was about 20 times higher than typical values. There was fish mortality and also low dissolved oxygen. Now, heterosigma this is not the first time we've seen heterosigma in the bay. In looking back at the microscopy data that goes all the way back to 1983, and then also molecular metabarcoding data that started in 2015, the colored circles indicate detections of heterosigma, and the red circles are in the 90th percentile of abundance for heterosigma. So it's here. It's in about 40% of all samples over the last 5 years from Lower South Bay to San Pablo Bay, but in particular, about 65% of Central Bay samples. And sorry, I forgot to mention, these are the different sub-embayments. So Central Bay is the connection to the Golden Gate. So we think that there's probably a high bit of exchange of heterosigma with between the coastal ocean, because we see more of these detections in the Central Bay compared to other embayments. And also in particular, there was a bloom reported in Richardson Bay, which is out by Sausalito in 2002, and there's a publication of that in the IEP newsletter back in 2003. So now looking a little more at the data, and we were able to also get field data in addition to the remote sensing. So these are maps from the our high-speed, high-resolution mapping boat. So chlorophyll is shown here as the color, and then the boat track is where that color is located. And you can see pretty good correspondence between the remote sensing and the ground data. So initially, when we pretty much got the first reports on August, just before August 10th, and we're able to quickly mobilize a boat to get down to the bay. At that point, the bloom had not extended south of Alameda. You can see there's low chlorophyll here. On the 17th, it had actually kind of dissipated a bit, like there wasn't that much, chlorophyll was not as high, and we thought that maybe the bloom and the event was over. Unfortunately, we were wrong, and a few days later, it just really, that's when it really took off, and you can see now chlorophyll is high, all the way down into the far extremities of the southern bay. And again, chlorophyll was on the order of 100 micrograms per liter, and so there were concerns then that as this crashed, that this would create a high demand for oxygen, which is exactly what happened. And so then things crashed on the 31st. The shoal moorings show that similar pattern, where in early August there's kind of this little bump, then things were lower, and this is probably a result of the tides just moving around where the bloom was concentrated. But then at this SHL site, things just really ramped up. Daily average almost approaching 75 micrograms per liter, and then a very rapid decline at the end of August. And the same pattern occurred for dissolved oxygen, where once that bloom crashed, dissolved oxygen decreased. And the target for objective is 5 milligrams per liter of oxygen in the bay. And you can see that it was below that for about 7 to 10 days, and even below 3 milligrams per liter for 2 to 3 days. And this, we think, was pretty much bay-wide that oxygen decrease occurred. So there was a large amount of fish mortality associated with this event. The first reports occurred on August 22nd. That's kind of indicated here by these green stripes. And then when we were out on the field a couple of days later, we observed numerous dead sturgeon and striped bass in the water. And what we think is that these first events occurred when oxygen was still quite high in the system. And so we think that this was probably a result of the ichthyotoxins that are produced by heterosigma, and not due to hypoxic conditions in the bay. And then with time, there was a citizen science web form, and let's see, a large amount of kudos and accolades goes to the folks who worked on the fish response. It was very rapidly, people put together a couple community science fish reporting forums, and it required a lot of coordination between the different fish groups. So we had the public reporting where they're observing fish kills, in particular that like basically all of Lake Merritt and Oakland went anoxic, and there was a huge, tens of thousands of fish died there. So this was the first major fish mortality event that's been documented in San Francisco Bay. And here's a couple pictures. It is rather sobering. I mean, these sturgeon can live over 100 years. So you've got, fish that have been alive for decades and decades that then succumbed to this event. And so in the end, there was about 250 reports, over 200 sturgeon. There's also been some research out there that only about 10% of sturgeon, when they die, they actually bloat and float to the surface. So the actual number of fish impacted could be potentially up to an order of magnitude greater than what we actually documented in terms of the sturgeon. Other thing, leopard sharks and bat rays don't float when they die, so we've definitely underestimated the mortality to those organisms too. So then what about the other component? We started off talking about nitrogen, and that's what we think is one of the drivers of this event. So this is the nitrogen panel below here, and what you'll see is there's a nitrogen and chlorophyll are inversely correlated. So at the beginning of the bloom, we had about 10 to 20 micromoles of nitrogen. And then, or sorry, this is in July. At the beginning of the bloom, it bumped up a little bit, but you can see here where there's low chlorophyll, where the chlorophyll is blue, the nitrogen's up to 30 micrograms per liter. Where there's high chlorophyll, that nitrogen has been consumed and it's very low. And that pattern continues so that by the 25th, when the biomass was at its highest, basically the whole bloom, the whole bay had no nitrogen left in it. Again, you can see the nitrogen tank there. Dave and SFEI have done some calculations, and they estimate that approximately 550,000 and 400,000 kilograms of nitrogen were used to produce many millions, over 5 million kilograms of organic carbon. And so in thinking about that utilization and kind of management implications, so in early August, we had about 900,000 kilograms of nitrogen in the bay. That's about a 20 to 30 day supply of those wastewater treatment loads. Then when the bloom happens, all that nitrogen gets consumed and transferred into about 6,000 kilograms of carbon. Oxygen goes up at that time, but then by August 31st, as that phytoplankton is then decomposed, we see oxygen decrease. And so the question is, what are the nitrogen levels? If we had sort of reduced standing stock of nitrogen at the start of the bloom, how much would we need to reduce that in order to have prevented oxygen from decreasing below 5 milligrams per liter? So it's potentially looking at about a total of 15 micromoles per liter, which is about half of what was there currently. So these are the decisions and the information that Dave and SFEI are trying to communicate to the regulators and wastewater treatment plant operators to try and prevent impacts such as this, such as the drop below 5 milligrams per liter. So overall, we still we have a number of hypotheses about what started the bloom. There's a lot of factors, like I mentioned, mixing, temperature stratification or temperature effects, light availability. These are motile organisms, so they can't swim up and down the water column. They're also susceptible to grazers and viruses. We have a pretty spatially complex bay with a deeper central bay and shallower south bay. So we're still kind of working through all of those components and don't have answers for you on that. But hopefully one day we will arrive at those answers. Kind of an update to that was in 2023, there was a return of heterosigma, which we were all kind of waiting with anxiously see if that was going to happen. So in late July 2023, reports began to trickle in of discolored water on Berkeley and Emeryville's marinas. Then a few weeks later in Seaplane Lagoon, here's some drone footage of that. Heterosigma was confirmed that was the organism blooming and causing discoloration at these locations, but it never really expanded fully across the bay and turned into the event from last year. And there was few fish mortalities reported, and Karen's going to talk about that after us. So that was good news, but again, we still have a lot to learn on this organism to understand why 2022 happened and what might have prevented that from happening in 2023. So that's the wrap up on the bloom. I'm going to kind of go quickly here and provide an overview on this new project that we got funded that we're excited about. So NOAA has a few funding mechanisms for HABs. One of them is the Management and Event Response Program called MRHAB. The goal of this program is to try and fund projects that aren't necessarily focused so much on kind of like cutting edge ecological understanding or technology development, but rather trying to mobilize and move technologies or proven devices from kind of research piloting studies into more operational and routine use by managers and by management community. And so in preparing this proposal, what we were trying to do was identify tools and resources that were available and think of ways that we could better implement them in San Francisco Bay so that we have more information and more understanding of the Bay to make the management decisions that need to happen. So it was called New Technologies and Coordination Framework for Integrated HAB Detection and Monitoring System across San Francisco Estuary. The proposal was led by Dave Sen, myself, and Ellen Priest at Department of Water Resources. Our other PIs were Ari Chelsky, Rafael Kudela, Tim Otten, Meredith Howard, and Tom Mumley. And then we had additional partnerships with San Francisco Baykeeper, Restore the Delta, and Cal State Maritime Academy. So a big team of people who are behind this and working together to develop these ideas. What's also particularly exciting is it's a 5-year project, so it's going to go on for a while, and we have a total of $3 million over the course of those 5 years. There was 4 objectives to the project. The first was to enhance existing monitoring data sources with new technologies and tools. The second was to then integrate all these different data streams more effectively so that we can deliver that data to managers via decision support tools for responding to and managing events. There's also a research component to fill data and knowledge gaps about processes that transport cells and toxins between the delta and the bay. And then lastly, the goal is to develop a coordinated HABs monitoring and management strategy across the entire San Francisco estuary. So currently there's been a fair bit of movement looking at monitoring strategies just for the delta, but there is yet to be a fully integrated strategy that kind of goes from freshwater to saltwater. So we're hoping that over the next 5 years, we'll be able to do that. This here is a figure showing the infrastructure that's in place or that we're hoping to deploy as a result of this project. And so you can see we're trying to really kind of be integrated, looking at cyanotoxins and cyanobacterial impacts from the delta into the bay, but then also the potential of harmful blooms from the bay making its way up into at least the more brackish portions of the estuary. So the first bit for enhanced monitoring, there's a component for work on remote sensing. So the goal here is to improve chlorophyll algorithms for both the bay and the delta using Sentinel-3 and Sentinel-2 satellites. There's also going to be new in situ moorings in the central bay to help with remote sensing calibration and also to expand the monitoring network for chlorophyll. The other technology we focused on is molecular methods, and so this is going to be we'll be working closely with Ben Genetics. So the idea here is to improve molecular assays for phytoplankton detection and enumeration in the Bay Delta. So we're working on 18S metabarcoding database refinements, improved qPCR multiplexing, and then also expanding 18S and 16S metabarcoding sampling into the Delta. So currently there's sampling for DNA, and then metabarcoding is done in the Bay, but not in the Delta. So we want to have a fully integrated and combined Bay Delta data set. We'll also be doing community science monitoring. This is going to be as part of through NOAA's phytoplankton monitoring network, and San Francisco Baykeeper will be leading this. So they're going to help establish a network of community science microscopists who will be collecting samples and doing early warning detection in the marinas and coastal areas that are hard to monitor with remote sensing or through the current other programs we have. The objective 2 was the data integration visualization. The goal is to telemeter the San Francisco Bay moorings. Currently they're not, so we only get the data back every month when a crew visits the site, but soon we'll be able to get that data delivered in real time. And then that will then lead to data integration and dashboards so that managers can pull up a dashboard and see the current status of all the sensors in the Bay. The third component is toxin transport. As I mentioned, this area from Carquinez Bridge to the confluence has been less well studied generally in the system. And so what we want to do is focus on this area and be looking for the potential of harmful organisms to be, again, moving out of the Delta and into the Bay, or up from the Bay and into the Delta. And so this will include both boat-based sampling and shoreline sampling. And also the shoreline will include looking at organisms, so toxins in clams or crayfish, as well as deploying spat. And then the last component is the management strategy. So this is going to be a multi-year effort to work with managers and stakeholders to write up a document that will help chart a course for what integrated and harmonized monitoring could look like. And the co-chairs for that are going to be Meredith Howard and Tom Mumley from the Central Valley and San Francisco regional boards. And we've already received letters of commitment from BACWA, which is the wastewater treatment plant, a wastewater treatment discharge organization, the State Water Board, Department of Fish and Wildlife, NOAA NIMS, BWR, Delta Science Program, San Francisco Baykeeper, and Restore the Delta. And you may be hearing more. We are going to be looking for other folks who are interested in participating in this group. And this will probably come together. This group will be meeting regularly, but the final strategy will be kind of coming at the end of the project. So, in about 4 or 5 years, that will be out there. And so when you put it all together, it's kind of a complicated, well, there's a lot of pieces which we're proud of what already exists. So all the blue dot squares are what's currently going on, and then the red shows what's going to be added with MIRHAB, so increased remote sensing, enhancing the water quality mooring, the shoreline, and phycotoxin sampling, supporting monitoring cruises. And the goal of all this is to feed into the dashboard for better decision support information, and then also feed into that monitoring strategy. So our hope is that in 5 years, the Bay Delta will be a more coordinated and communicative place for HABs monitoring response, and we'll also have more data available to inform decision making. So yeah, so that's what we are up to. If there's time for questions, we'll be happy to take them, but otherwise feel free to follow up with me over email as well. Thank you.
Thank you so much, Keith and Dave, for that information. There's about 3 minutes left for questions if we'd like to. Anyone has some?
I did see Hans has his hand raised.
This is Hans.
Go ahead, Hans.
Hey, Keith. Nice presentation. Hey, I had a question about the about the termination of the heterosigma blooms. You know, the graphs, by the way, are very useful to look at that. But if you look at the graph, go back to the graph where you look at nitrate and the, yeah, that one right there. So the nitrate runs out, which, you know, is not surprising, but then the bloom keeps going, and then it crashes very quickly. Do you have any idea why that was such a sudden crash? Was there something about the physical conditions? Did it get attacked by viruses? Any hypotheses?
Yeah, no, that's a great astute observation, and yeah, exactly. Like, so, and we do and see it continue and then crash. And Dave's the one who's looked the most closely at this. So Dave, do you want to take that?
Sure. Hi, Hans.
Hey.
Well, Keith is showing just a subset of the mooring data we have here. One of the things that was really noteworthy is that even though the elevated chlorophyll started at different times at different locations in South Bay, it all terminated at almost exactly the same time. And the oxygen dropped off abruptly, oxygen drops were instantaneous almost, or suggesting that it was pelagic respiration as opposed to benthic respiration. And one of the things that we're exploring is, as nitrate decreased, there's still some ammonium left there, but the discrete samples we found showed that ammonium was quite low once nitrate was really low. You can sort of see if you look closely or imagine that there's a change in the slope of the chlorophyll increase right around the time that the nitrate drops down. And so a change in perhaps nitrogen limited growth rates. And one of the ideas being that something needed to allow this bloom to take off. And there's the physical factors that allowed it to grow. And there was potentially the alleviation of top-down pressures, either from grazers or from viruses, as you suggest. And so one of the things that we're curious about, and we have some samples set aside for this, is whether it was actually the viruses ultimately catching up and leading to cell lysis, abrupt cell lysis all around the same time.
Yes. Cool. What about the weather? Was there like something happened in terms of a storm or something, or some kind of physical perturbation that might have caused it to crash?
Nothing obvious about the led to the crash. There were some changes over the course of the 1 month that we're exploring whether they could have contributed to or slowed the spread at different times and places, but nothing towards the end of August that would have, let's say, we didn't have any 5 days in a row of cloudy days, for example.
Okay, great. Well, interesting question. I think the managers are going to want to know that too, I would guess.
Thank you. We are out of time for questions unless there's something really pressing that could go quickly. So we'll go ahead and move on the agenda. Thank you so much. Really fascinating presentation. And Keith, I'm assuming that with your MIRHAB project that you all will be updating CCHAB from time to time on that as well.
Yes.
Okay, great. That's
great.
wonderful.
All right. Thank you, everybody.
Thank you. All right, our next item on the agenda is a 2023 Heterosigma bloom response at San Francisco Bay with Karen Odkins at CDFW.
Hello.
Karen, go ahead.
Yeah, let me share my screen here.
When Keith comes to the water boards to set up the HAB team, and then he
Oops.
leaves to go.
Make sure
to
you've got your
USGS.
mute on, folks. Thank you.
Can you all see my full screen?
It was there momentarily and then disappeared.
Okay. When I press slideshow, you can't see it?
No.
Okay. All right. Let me try this way. Still no?
It was there for like a moment when you first got on, but it's not there right now.
Okay. Let's try it this way.
Oh, there we go.
Is that full screen now?
No, it's
Okay.
showing all your slides.
How about now? Is it full screen?
Perfect. Thank
Awesome. Okay. You guys can tell I've been working with Teams for a while here. Okay. So yeah, thank you so much to Keith for setting me up so well here with talking about all the water quality aspects. I'm going to be talking about sort of the CDFW response to the fish kills that happened this year. I was not at CDFW in 2022. But we did, from what I've heard, made a lot of kind of improvements from learning from 2022, and I would love to share with you guys about that. So as Keith mentioned, I just have a really quick slide. So 2022, there was this huge bloom, many fish died, and it was just a really tough, sad time for CDFW and all the outcomes of that. And it was really hard to keep kind of track of everything happening because it was really a unique situation we hadn't really faced before. So things were not necessarily, or the response wasn't necessarily in place in the way that we had it in 2023. So we spent kind of the year in between really working on trying to improve some of those. So I will talk about that now. So in this year, we had a field response. So we had a field team that was ready to go out, and they went out 4 different times throughout the bloom to different locations. Those locations were kind of based on information that we had from last year and where the bloom was really bad, but also from where we were seeing those satellite images like Keith showed, where we saw really high chlorophyll levels. And so our teams went out to Berkeley Marina, Berkeley Aquatic Park, Lake Merritt, and then also the Northeast San Pablo Bay, like you can see in those red circles here.
Thank you.
But obviously our team couldn't be everywhere at once, and we only had so many resources to send folks out and divert them from their other jobs. So we utilized the iNaturalist platform. And I think Keith kind of touched on the fact that in 2022, there were a bunch of different kind of platforms that people were using to try to collect that community intelligence and data that they were collecting. And so then on the back end, when we were trying to analyze it, it was really difficult to combine that to see where there were overlaps in data, where the same fish might have been reported twice, that kind of thing. So we really wanted to make sure that all of the data was being collected in one location. So we decided to do that on iNaturalist because it is just so easy to use. We had 112 members of the public join the project, which was really great. And we had 24 of those people actually submit observations. And the way that iNaturalist kind of works, if you're not familiar with it, and the way that our project in particular worked, is that you join the iNaturalist platform, you join our program or our project on it. And then if somebody wants to be admitted, okay, if your project or if your observation in particular meets our criteria and meets our filters, then it gets pulled into our project database. So the filters that we used this time around was that the animals had to be dead, they had to be in the San Francisco Bay Area, they had to be within kind of the date ranges of when we were seeing the HAB, and they had to be, relevant taxa to that which would be affected by the HAB. Okay, so of course, iNaturalist data, just like all data, has lots of limitations. So these are all observational counts of what people went out and saw. We were able to spatially QC it. So we looked at the data, tried to see where there might have been the same fish kind of or similar picture that was reported near each other. And then we were able to kind of pull some of those out. And then we also did QC for taxonomic IDs and tried to get those more accurate. Of course, as you can see in this picture, some of what we were seeing in the field were just, really decomposed species or specimens. And so those, of course, we're not really able to ID those to a fine taxonomic level. In addition to that, like Keith mentioned, this is going to be a really low estimate of the impact because some casualties don't actually make it to the shore at all. They might sink, for example, like Keith mentioned, and some of them, some of the area was not even looked at either. So, some of this shoreline may not have been counted. So what you're about to see is based on incomplete and imperfect data, but it's kind of the best that we have to go on. So here's the complete species list you can see on the right, or the kind of taxonomic list. Some of these are pretty crude at the top and then they get down to species. But as a sort of summary here, you can see we had 1 Chinook salmon that was reported, 23 sturgeons, that's a lot less than we saw last year, crustaceans, and then of course ray fin fishes and aclasmobranchs. We had 88 total organisms that were reported on iNaturalist. Not including, of course, the ones that we removed for QC reasons. One thing that was not added to iNaturalist was that there was a mussel bed that was affected, and that probably was thousands of individuals, but it's really hard to estimate those numbers. So that was not included in those counts. Spatially, here you can see the satellite data on the left that Keith was showing with the chlorophyll. And then on the right, you can see a map of where our observations for iNaturalist were located. You can see that those locations, of course, were also highly influenced by where we actually went out to send field teams to sample. But we also have other observations all around. But those were really concentrated around Berkeley area and San Pablo Bay area this year. And then in terms of the timeline, it really seemed like in August, it was kind of ramping up, and then it kind of had a lower, kind of tapered off here. And of course, that's also influenced by those observations when we sent people out to go see the observations. And lessons learned here. So I think this year we did really well with interagency communication and also including, the stakeholders and nonprofits that were involved. We had a meeting earlier in the summer before the bloom happened to kind of talk through what a bloom scenario would look like, how we would handle it, and get folks who were going to be the lead from each agency already figured out. So we had kind of an email listserv ready to go, and we're able to meet really quickly. In terms of supplies and field teams, we had folks identified at CDFW, and I think nonprofits also had some folks that were ready to go this year, which was really great. We also had, at CDFW, we had a collaboration already figured out with our fish health lab for autopsies that came up. And we also had a contract this year that's new in place for the Greenwater labs and vent genetics. So that was all positive improvements. Unfortunately, this year we didn't have any samples that were actually collected, mostly because the samples that were found, like you can see in the picture of the bat ray on the right, the samples were not actually fresh enough to really collect for autopsy. But in addition to that, we had some real permitting questions that we were not able to really quickly figure out. People were worried about moving listed species, rightfully so, and if they weren't sure if it was listed, they didn't want to be moving them because that's illegal. So CDFW staff, though, we now know can move listed species without any issues. We have that ability, and I am currently working on solutions for partners, and we are getting closer there. Upper management is talking about how best to do that. So yeah, that's my little update for you all. there's any questions, I'm happy to take them.
Are there any questions for Karen? Karen, I have a question for you.
Yeah.
So that was very interesting, and thank you so much for that presentation. And it's awesome to see CDFW out there collecting this data and being so transparent about it. You might have said it earlier in the presentation, I missed it, but how are you determining that these are HAB deaths or mortalities of the fish? Is it more that it's a DO issue, or are you actually seeing toxin levels that are at some mortality level for the fish?
That's a great question, and we are just basing it on time and location in this case. There's no really way to know the difference. I was hoping to get a little more insight on that by being able to send in samples for autopsy and for lab analysis, but since we weren't able to get any samples in that this year for that,
right,
that kind of prohibited that. But hopefully in the future that's something we'll have a little more information on.
Has CDFW set basically kind of thresholds for fish? I know there's been some studies that actually had some presentations on that in with CCHAB of just what levels would be fatal to fish. So have you started to develop that? I see Shannon has her hand raised as well. She might want to jump in on that too.
Yeah, Shannon, if you want to, you can.
Sure.
I can also answer it if you prefer, whatever works.
Sure. Just one thing with Heterosigma akashiwo actually translating to red tide. It was first identified in Japan in the Northern Pacific. There's no known toxin compound that's an actual toxin that's associated with this particular biflagellate. And so what the understanding is that when the organism, the biflagellate in this case, interfaces with the breathing mechanisms, the gills, in particular of these fish, it's thought that maybe there's a cellular reaction such that it creates kind of a surfactant type compound that gums up the gills so that it really suffocates them. And so it's called a fish toxin, but it's not really a toxin per se that we have yet to identify, not just us, but in the many decades that these kind of blooms have been observed in other parts of the Pacific, that there's not a compound specifically that's been identified, but really more of this kind of surfactant type cellular reaction that it compromises the air exchange for these fish, resulting in their death. And really, it's catastrophic to our sturgeon population. That was perhaps the saddest component of last year. So it behaves in all the other ways that a HAB does, that it can cause the environmental impacts and compromise the dissolved oxygen levels. And because, as Greg or as Keith had pointed out, it is highly active. It can bloom quite quickly, especially if there is a warm water event such that you have the stratification of the water and high sunlight penetration. They really can take off as far as concentration. So those are some of the contributing factors for 2022. But as we saw in 2023, thankfully the persistence, concentration, and spatial impact of this bloom was markedly less. Similar impacts as far as the types of things we were seeing with fish deaths, but certainly not on that scale. So again, the mechanism of action is still being studied, but it's really thought to be associated with a reaction that with the fish gills such that it creates a sticky surfactant type compound that prevents them from having more effective gas exchange rather than a toxin. I had a couple weird ones today, that and Noctiluca, so very good to know file.
Thanks, Shannon. That was really helpful. And also, I mean, that's partly why we thought autopsies in particular could be really interesting, because we might be able to see some of that.
That sounds great. And thanks for that question, Shannon. We are actually at break, and we have a break until 10:50. If I know I saw a couple hands raised, and I'm
Yeah, I'm happy to stick around during the break if
okay.
you want to ask your question, but everyone else can go take their break.
All right, folks. Thanks. See you back at 10:50.
Yeah, Karen, can I ask you my question before? I don't want to hinge on your break too much.
No.
But yeah, I think Shannon brought up a good point about, the the heterosigma doesn't have kind of like a toxin in the sense that we think of like, you know, microcystins or domoic acid. But I think regardless of toxin, like the international like HABs kind of community that tracks like blooms of international significance, they even just kind of include like hypoxia by high biomass events as like a harmful algal bloom, regardless of like if there's a toxin. So
so I guess if you, depending on where you cut off that line of what is HAB related, right? I mean, right.
Yeah. So it's probably, you know, even depending on what I think heterosigma has well documented kind of ichthyotoxic compounds. But I think your approach of just saying, you know, we have the high biomass and then here's the time frame, because it's even with like your autopsy, like necropsy stuff, like on the sea lion side, stuff I've kind of been working on with like on the marine side, it's really hard to like say, unless it's a really acute thing, like even with like testing tissue toxins and that sort of thing, like that was the thing that killed the animal. It's usually kind of like a lines of evidence approach anyway. So I like your approach, kind of looking at the, you know, this is the time frame that, you know, outcomes razor, right? It's almost certain.
Totally.
And
I think you can kind of see that in that like temporal graph too, like not many, oh wait, the bloom happened, and you can see that it tapers off afterwards too. So it kind of, makes sense in that way.
I really like the iNaturalist approach. I have all kinds of like offline questions to ask you about.
Cool. Yeah, I was just learning how to use iNaturalist for the first time. I hadn't really used it myself before, so this is steep learning.
Thank you. I'll let you
take questions. Yeah. Thanks for your questions, guys.
Hey, Karen, I'm impressed you had 112 people interested in participating. That's fantastic.
Oh yeah, it was awesome. We were able to do a lot of outreach with it, and a lot of just like the community group members who had participated last year in iNaturalist were really awesome at like bringing people back from last year. And we were just really, you know, from the beginning, the press release was like, help us in this way, please. So I think having that coordinated in advance was really helpful.
Yeah, we have a Clear Lake fish kill project on iNaturalist, and
we
have some people who participate in that, but it's not near the numbers that you're seeing. But then again, we haven't, since we put it together, there really haven't been too many big fish kills on the lake.
Oh, that's so cool. If you want, Sarah, I would love to like do some outreach for that if you're interested.
Yes, I'll send you.
Send me the link. And also I didn't know about it, so that's really cool. I'll keep an eye on it and join it myself.
Oh, thank you. Appreciate that.
Yeah.
And I am a toxicologist in the fish ecotoxin water section, and so we shall proceed. I'm going to go ahead and turn off camera. There we go. All right. So again, just named all the agencies that participate. All of our emblems are here on the right. We receive notifications for potentially, and that's key, potentially HAB-related human and animal illnesses. We coordinate responses for individual cases. Each case, as appropriate, is assigned a tracking number. For a human, it's largely led by CDPH folks. If it's domestic animal, a WEHA takes lead. That's most represented by dog cases. Fish and Wildlife goes to, of course, Fish and Wildlife, so that's Karen. And then response and agency coordination, as well as site-specific efforts to collect environmental data, particularly through our regional HAB coordinators, is headed up by Water Board. We essentially come to a consensus determination on which illnesses are HAB related. I'm going to talk a little bit about how we do that later. We investigate these HAB-related cases and complete the documentation for statewide tracking for California, as well as submit to CDC's database, which is OHABS, and that's One Health Harmful Algal Bloom System. So for data collection, these are the types of information that we're looking at. Obviously, we don't have everything for every case, but these are the general categories that help us gather the burden of evidence to see if we can make a determination as to whether it's HAB related. There is a possibility of an event being HAB related, but we have insufficient information, either lack of environmental information. We can't get in touch with the reporting party after they made their initial report. There's just not enough information from the symptomology. There might be confounding factors, other illnesses or pre-existing conditions in play, that all of these things can come together to make it difficult to make our determinations. But generally, this is the information we're interested in. And when we do report these to CDC's OHABS, again, that's that national database, every event or illness is accompanied by an environmental form. So we capture the information that we can, what was visualized, what did we see, smell, etc. And then if there's any laboratory data findings, microscopy findings, and then of course the appropriate illness report, whether it be human or animal. Illness determination is done by the committee, our work group of the 4 agencies. Really, for it to be HAB related, you need to have sufficient information on the illness and the symptomatic individual, or in the case, animals, the signs. Onset and type of signs and symptoms, are they consistent with what we know or has been understood to be associated with a HAB exposure? Example, symptoms that may arise from an inhalation or a skin contact dermal or an ingestion type exposure. Sampling results and observations that provide sufficient evidence of either the presence of cyanobacteria and or the cyanotoxins. And are those consistent with the symptom types and severity? We work with water body managers, and especially a shout out to all of our FHAB coordinators. You heard from them earlier this morning, to post HAB related signage. We do have signs available in both English and Spanish for statewide distribution where appropriate. When it's not HAB related, again, it may not be that we have a definitive rule out that we know exactly what it was and it's not HAB related. In some cases, we are fortunate to make that determination, but it's not always the case. So if there is insufficient evidence to really meet those CDC definitions, which I'll get into in a moment, then we determine it's not HAB related because there's insufficient evidence to say, yes, it is. It does not meet the burden of proof. So it's not necessarily guilty. It's just we can't make a determination definitively. So this is oftentimes our most frequent default. I will say, however, with the increased stable of experts that we're coordinating with, not only in the work group, but also with our expanded partner network, and then our new resources that we have online, particularly with our laboratory relationships, and then with CDFW, with the onset of Karen joining the group and having expanded laboratory capacity, we are able to make definitive determinations where previously we were not. So we're moving in the right direction as far as resources. Illness onset and symptoms not consistent with HAB exposure. During the time of COVID, we had several people report respiratory illnesses after recreating in a water body, and then they went home and tested positive for COVID. So these kind of things do happen. Another more likely cause of illness is identified, such as like a COVID or a flu test. Potential exposure only. So there was no illness reported. People were just concerned. They either saw visual signs of what they thought were a HAB, or there was in fact an advisory in place, but they were concerned and they might have made a call to either through our work group resources, through the HAB portal, or maybe even to Poison Control, another one of our partners. Even if they weren't experiencing illness, sometimes they will make a preliminary report just to get more information. Of course, with any of these endeavors and investigations, you're going to have several constraints and challenges. Timing is critical, though. That's the most important element. Once a report comes through, we really do our diligence to try and get in contact with the reporting party within a day or so, because we need to make sure that we are sending representatives out into the field to sample these water bodies and make trained eye observations on site if that's appropriate. We also want to make sure that they're getting the resources they need, particularly for people who are reporting dog illnesses, that their animals are getting access to veterinary care, and any guidance we can provide with our support sheets that we have for both veterinary and human medical providers. A couple other things too, that this is really something that's rocking the HAB world, is that we are challenging assumptions and kind of the dogmas of what we understand HABs to be. In fact, US EPA had a recent webinar presenting some of the information that was shown at cold water HABs and cold weather HABs events that were happening even at elevation in Europe and in different parts of the United States, that some like it cold and some like it hot. So again, really pushing our critical thinking skills and understanding to think outside the box a little bit about, is this a HAB event, even if it's not necessarily meeting the environmental conditions that we know to be consistent with the likelihood of a HAB. Sometimes it's a challenge because the water bodies where people were recreating have private or restricted access, including like at a golf course or a resort type situation, a gated community and the like. Limited analytical options for biological samples. And then of course, it's always challenging to know which toxins to test for. And there's other toxin classes for which we don't routinely monitor. Chronic illnesses, this is something that really falls outside of our jurisdiction. However, we have had a handful of cases where even though somebody had a pre-existing chronic illness that wasn't necessarily associated with HABs, their recent acute exposure or short-term exposure to a HAB event caused an additional set of symptoms and exacerbated the kind of inflammatory response of their pre-existing condition. So just because someone has an underlying illness doesn't necessarily mean that there isn't a HAB related illness on top of it for the short term. So, but this is something we tease out in our investigations. So we don't just do this willy-nilly. There are actual CDC guidance definitions for these cases, in this case, humans. So there are 3 levels to note. So when people are asking questions about how do you know it's HAB related, what is the burden of proof? Is there a way to kind of qualify that when you report it to OHABs? And there are. So there are certain definitions and requirements that have to be met in order to call it suspect, probable, or confirmed, for example. And you can see clearly on here which ones meet which criteria. And then of course, on the animal side, same situation. Animals are a little bit more challenging, of course, because not only do they not have voices for themselves, but they don't always read the protocols, right, and know to report. So, but you know, coming from a veterinary background, that's certainly been my area of you know, what I bring to the group. And so it's really helpful, I think, to understand how different animals interact with their environment. What are some of the behaviors that might make them more or less likely to have access or exposure to a HAB? What is a normal behavior? What's not? Those kind of things. So we really use our situational awareness as well as what we know about the species in play and how they behave. And this is why it's such a value to have our CDFW component as well, to really inform us on the wildlife biology side of things. So again, this is a very busy slide, but the key element here is that minutes to hours, that's when exposure can occur, as well as symptom development, and then symptom onset or even death. And especially in the case of neurotoxins, we've seen dogs drop dead at the lakeside. In fact, that happened recently in Lahontan, so region. So this is still a very real and tangible concern. And so response times and really getting those accurate narratives in the early hours to days after a potential exposure event is really critical to our information, so that people can report accurately, and then also that we can have an opportunity to capture the environmental data that's really critical for our investigation. So what are some of the highlights that we've been doing this year? We coordinate, again, ongoing relationship with the California Poison Control System. We've also had several discussions where they have updated their case coding such that they are capturing more potential illnesses, including those that might be foodborne in nature. So potential foodborne HAB-related illness. And then once they get permission from the caller to see if state health officials, if they're willing to speak with state health officials, those cases get uploaded to CDPH and OEHA, and then we follow up directly with those reporting parties. In some cases, we're able to identify what is the likely scenario. May not be HAB related, but one that we get not too rarely is scromboid fish toxin. And really, that's not a toxin. It's a histamine response that is caused in the fish that are not kept at the proper cold temperatures and are not processed in a timely fashion from the time they're harvested out of the water. And so when people consume them, particularly in like a sushi or a recreational harvest type situation, they can have a what kind of sounds like a panic attack / anaphylaxis, but it's really not, but they're having a surge of histamine in their body that can cause a pretty upsetting experience with heart palpitations and flushing of the skin, much like you'd see with an allergic reaction, and that can be very upsetting. And in those cases, they need to be forwarded to the proper health authorities at the local level so that an investigation can take place. They can analyze meal remnants if they exist, that kind of thing. Other opportunities would be other sources of foodborne illness, just to send them to the right authorities so they can be investigated appropriately. Another one was where we had a potential illness due to HABs at a marine beach in Southern California, and it turns out that there was a high fecal bacteria warning at this beach, and people were not supposed to be swimming, and they did so anyway, and so ended up having secondary symptoms due to those exposures. And so we had a opportunity to share those resources with them, have them, put them in touch with the right websites and beach officials, particularly in Los Angeles County, to know the information and the status of their beaches before they go out and recreate. So again, we're not always looking at HAB-related cases, but because we've had these greater outreach opportunities and expanded coding relationship with the California Poison Control Center, we're better serving the public by putting them in touch with the right resources. So those are some of the elements that we contribute to as a state service. We provide biweekly illness reports to water boards. So all of you regional HAB coordinators should be getting those basically updates from me every other Friday. They might be late Friday night, but you tend to get them. If there's any training opportunities or key webinars or other resources, brand new paper that was published that I think would be really helpful to you folks for HABs, I add those in. So I try not to over communicate with the HAB coordinators because they're very busy unless we're working up an individual case, but do keep an eye out for those, because you'll see on the table that I add in those emails every other week that you'll see which regions are impacted, what the nature of the case was, and what is the status. Did we make a determination? Yes, it's HAB related. No, it's not HAB related, or it's IP or in progress. So those are really, that's kind of a snapshot to keep you in the loop. I try to highlight which regions every other week are being impacted. So I'll put those in parentheses. Not that I want you to ignore my emails, but if your region's not called out at the top, then maybe you can just read that at a later time. So try to give you a little bit of shorthand for that. Currently, to date, we have 8 human illnesses, 3 dog illnesses, 2 dog deaths, 4 fish kill events, although a couple more are in progress, so that I anticipate will be expanding. And right now we have 5 marine mammal stranding deaths, but that's only those with final numbers. So as many of you are aware, there's been a significant domoic acid event that happened in late May through June and July. We have to date probably an estimated 1,000 marine mammal strandings that are pinnipeds. So those are mainly California sea lions, fur seals, and I think there's 1 or 2 other smaller pinniped populations in there, and then over 100 dolphins. So those are very sad. Our partners with NOAA and all of the Marine Mammal Response Network in throughout the state, they are getting their final numbers to us before the end of the year. And so we will be writing up those reports as domoic acid related stranding events. Many of them had either survived and then subsequently were euthanized. Some were able to be hospitalized and returned back to the ocean. So those will be captured. There will be a lot of paperwork in our future after Thanksgiving for the team, but we will be capturing those events. So this 5 is not representative of what's going to be showing up for California by year's end, unfortunately. So one of the key questions we get, and I see a lot of chats piling up, but please stop me, one of the co-chairs, if there's something I need to answer right away. So flag me down. For sample case determinations, I thought I'd just illustrate 3 quick studies. People ask a lot of questions. How do you make your determinations? Obviously, we don't always have every piece of data that we're interested in that would give us a clear-cut case. Here's case study 1. It was human illness, 2 minor children. It was reported at a lake that did have a caution level advisory. The mother reported respiratory symptoms in 1 child within 24 hours of recreating at this location, so possibly HAB related. It was worth investigating. Environmental evidence is planktonic bloom cyanobacteria were present and toxins were detected. So of course it's looking like it's fitting the mold, right? But we made a determination that it was not HAB related. Why? Well, as I said, there were 2 children recreating, and they weren't seen actively ingesting the water, but they were seen with dermal contact. There was evidence of a bloom, but not immediately in the area where the kids were recreating. So the first child developed respiratory symptoms consistent with what you might see with an allergy or a cold within 24 hours. And then within 5 days of exposure at this water body, the second child who was recreating at the same time started to manifest the same symptoms. And so it appeared that there was a communicable illness component, of which HAB-related illnesses are not. And the second child developed very similar symptoms. And then there was some background information from the mom that there was a known virus that was going around through the children's school. And so we determined that this was not HAB-related because with the type of exposure that they had, we would be more inclined to anticipate GI symptoms and dermal irritation, and those were absent. And the fact that only 1 child had the respiratory symptoms and the second child developed them 5 days after exposure suggested that there was a communicable disease element like a common cold. So we decided that one was not HAB related. Second case, dog death, and we have seen these a few, particularly in our benthic environments. A dog was recreating in and out of the water at a place where there was no clear evidence of a bloom visually, although it's one that had historical benthic blooms. With the environmental evidence, there were some mats that were observed offshore. Cyanobacteria was present in the water at very small amounts. No toxins were detected. The animal was coming out of the water over a couple of hours, was observed drinking the water in copious amounts, and then also was observed eating some dried algal material on the shore. Within minutes of being observed drinking the water and eating this dried algal material and having some residue of algae-like material on the coat, the dog started to stumble, had trouble walking, appeared to be out of breath, was alert, but having some cognitive challenges. And then within the next hour, the dog succumbed to respiratory arrest and died. So there was certainly evidence of a neurological component. And with the type of cyanobacteria present, there were 3 different kinds, and they were all associated with known producers of a variety of cyanotoxins, including anatoxin A and saxitoxin. So we've seen this a couple times in the benthic events where they're kind of the sneaky blooms, dogs are in and out of the water and then are dead within 1 or 2 hours of recreating, and the neurological symptoms are present. So even though toxins were not detected, even though an algal sample was analyzed, we know that there can be orders of magnitude variability between algae mats, even in the same vicinity. So you can have some that are non-detect, or you just detect the cyanobacteria and no toxin, and then others that can have orders of magnitude higher levels of toxin. And even adjacent mats can have one type of toxin, another mat close by within even feet away can have multiple types of toxins. So this one we determined was a HAB-related death because of the significant presence of neurological symptoms, no other pre-existing conditions. This was a healthy young dog that would suggest that there would be cause for concern for some other contributing factor for a CNS event, central nervous system. And lastly, with the fish death, this is something that was really a collaboration between multiple partners. There was a large fish kill observed. This was in Central Valley. However, only one species or one type of species was impacted. These were the goldfish and carp. And so it's quite unusual for a HAB event to have only one species impacted when you know that it's in a location where there is quite a lot of biodiversity, several types, right? Including more sensitive species, if you will. For example, if it had been a dissolved oxygen event, you would anticipate a lot more of the sensitive species being represented in this large fish kill versus just the goldfish and carp. And so there was also, this was a lake that was known to have repeat HAB events, very severe ones, chronic ones that lasted over a long period of time, and ones that would morph into various phases of representative cyanobacteria and different types of toxins. However, you have to go with what you see at the time of the event. And this is why it's really, really critical not to make assumptions about your water body, even if you know it, because there was no visible bloom indicators at the present time, and there was no cyanobacteria present. And yet only one species was impacted. So we took a step back, and I remembered a case study that was similar, and it was the cause of death was determined to be koi herpes virus disease. And so through our partnership with CDFW, they collected these samples and sent them off to the lab, and UCDA has confirmed that 100% of the samples submitted were positive for koi herpes virus disease. So this was not HAB related. So again, it's in each of these scenarios, it's important not to make your assumptions and to use that critical window of the first, couple days to a week to get those accurate narratives, follow up with the reporting parties, and get as much information as you can to have confidence in your decision. Just a quick overview. I remember I told you we were already at 22 cases to date, and that's going to grow exponentially, particularly with our marine mammal strandings. This is where we were in 2022 with 20 human cases, 3 dogs, 7 fish or wildlife incidents. And of course, we always welcome partnerships with everyone. So if there's a way that we can help you with outreach events, if there's a conference we can assist you with, if you're preparing some sort of an update or want to share some details that would be appropriate, if there's some way that we can highlight a unique case study that you found in your region or experienced or think people could benefit from historically as we go forward, especially at the state level, we'd be more than amenable to talk to you about that. We do meet every other week officially as a committee. We oftentimes will have check-ins via email or the occasional call multiple times during the week. And our caseload ranges anywhere from 1 or 2 cases ongoing to we've had as many, I think, as 12 or 13. So we don't have a bloom season in California because we have blooms year round, including in the cold season. High elevation, low elevation lakes, estuarine, freshwater, marine, we take them all. So really encourage everybody to continue to spread the HAB mission and messaging that HABs continue to be a persistent problem, but that we can better equip people with information on how to check the HAB map, report HAB events, check their local listings, as we used to say, for what conditions may be present at water bodies they frequent, and that there are resources and guidance available in the event that there is a HAB-related illness suspected. So with that, I'll take any questions.
Thank you, Shanna. That was really good. I think I saw Jamie's hand up.
Yeah, please.
Well, actually, Shannon, you answered my question because I was like, oh, there were certainly more than 5 sea lions that stranded this summer. But yeah, I guess I'm just curious, informational, how do you, what is the kind of benchmark? You said that there's 5 confirmed, and then, we think that there's up to 1,000 animals involved. Like, are you looking at each of these cases individually as the work group, or are you working with the stranding networks and they kind of make the case determination and then you intake that information to get those phone numbers?
Excellent question. The short answer is both. So
Okay.
historically, before this year, we've had stranding events and, upwards of 5 to 10, maybe an event. Sometimes there's 20 cases. And so we work directly with the Marine Mammal Network that responds to these. Most of them are in hospital or, follow these animals. And based on their either clinical assessment, based on symptomology, the location where they stranded, was there an active bloom at the time? Because we do have that environmental data as part of our partnership with the groups. And then also they do some confirmatory laboratory testing with, analyzing feces, presence of domoic acids detected, et cetera. It's mostly based on clinical syndrome. And since these folks are so acutely aware of how these DA cases present, we rely on their expertise in making those determinations. So we get a preview, mainly it's a WEHA, a spreadsheet kind of from behind the curtain that, when they have their preliminary data ready to go, they will share that with us. Look, these are going to be the reports forthcoming in the next update that they send out every other month or so. And so when we get those case numbers, those are actually confirmed DA related illnesses and or euthanasia/death events that are determined by our Marine Mammal Stranding Network, largely in Southern California. And then those get sent up as individual cases up to us. And then we report them to OHABs along with the accompanying environmental data that's provided through Scripps and all of our other partners in Southern California.
Very cool. Yeah, thank you for that description. And I feel like I'm going to email you offline because I have many other questions, but they're much more Marine Hab kind of focused. I didn't want to divert the meeting today. So thank you.
Yep.
Oh, cyanobacteria.
Oh, if you could mute your line, the person who we can hear you in the background, I'll go ahead and mute you. Any other questions for Shannon? All right, thank you, Shannon, for that.
Thank you.
Excellent report. We are just have a couple more things on our agenda. One is the planning for 2024, and I believe that Jamie already posted the 2024 meeting dates, but I'll go ahead and put it in the chat right now. So our next meeting is our larger annual meeting, which gives more time for the regional HAB coordinators and the work group and the work groups to report out on their work. So we'll be going from 9 to 2 on January 31st. Then we've got April 24th, 9 to 12, August 14th, 9 to 12, and November 20th, 9 to 12. As far as we know, these are going to be virtual meetings. So just look for the Zoom links on the agenda. Please note, we're not doing this by Teams links, at least for this coming year. We've got Zoom links coming out. So always look for that. As I mentioned earlier, there are going to be co-chair nominations in the spring meeting. So that's in April for Jamie's and my seats. If they're so think about if you're interested in that and running for that. Are there any other for the co-chairs? Are there any other planning things that you want to discuss or have announcements about?
I just ask an open question to the group of, we recruit speakers and look for specific topics. I heard earlier in the meeting there's some interest in maybe trying to recruit some talks about wildfire impacts on harmful algal blooms. Are there any other topics that folks would really like to hear about in the coming kind of year of meetings? And if putting you on the spot, if you think of things that you'd like to hear about, please go ahead and feel free to email the co-chairs, and we see if you see any interesting papers or anything like that, we can recruit speakers based on the community's interests. So we're always open to suggestions. I think besides that's the only thing I had on my mind, Sarah.
Thanks for bringing that up. Karen, anything with you?
Nope.
All right. Any other topics, planning topics or questions that the group has? If not, we'll move into just open discussion and wrap up. Are there any comments or topics or announcements that people haven't had a chance to make that they'd like to make now? Just raise your hand or put it in the chat. Okay, Christine, Joab, but further updates on research looking at inhalation exposure. I was thinking of the same thing, Christine. Yay. I definitely want to get some updates on the research going on in the, isn't it in the Bay Area for inhalation exposure? I think we hit all of the questions that were in the chat, signage with Marissa and Carly, if you need signage. The presentation will be on the YouTube page of the California Water Quality Monitoring Council. And we'll have we'll be updating the website with the new dates of 2024 meetings. All right. Anything else? All right. Thanks, folks, and thanks for your attention and time and questions, and to all the presenters today. And we will be posting this on the YouTube page within the week. And thank you all again, and see you next time.
Thank you.
Okay.
Thanks, Sarah.
Thanks. Bye.